On Friday, we introduced the idea of gene regulation. You have your little note sheet, hopefully, about sort of the differences, different categories of gene regulation. We're going to go through, we'll go over the trip operon again. We'll talk about the lac operon today. Okay? And operons, what is an operon? Nick, what's an operon?
It's too early.
Nick says it's too early. Amanda, what's an operon? Sarah.
It's an entire stretch of DNA that includes the operator, the promoter, and the genes that they control.
All right, so it's a cluster of genes, including its regulatory elements, and they're found where? In what? All right, they're found in DNA, but in what type of organisms?
Oh, we
In prokaryotes, okay? They're found in prokaryotes. And all of those genes that are regulated by the operator sequence, its regulatory elements, they are usually what? What can you say about them? They're usually activated or somehow activated. Okay. Are they related? Yes or no? Are all those genes that are within the operon, are they related? Yes, kind of, sort of. Remember what the operon looked like? Right, if we were to draw it out, we have an operator sequence. Right, that's the operator. And then we had several genes. Right, and they were TRP, E, D, C, B, A. Right, and each one of these genes coded for an enzyme. Enzyme 1, 2, 3, 4. And each of those enzymes they catalyzed what? Look it up. Look it up. You have your notes in front of you. What did they do? All right, they were important in the formation of what? In the production of so specifically the TRP operon. Where the genes of the TRP operon were important in the synthesis of tryptophan. Right? So you went from precursor molecule, subsequent steps, 1, 2, 3, 4, 5, ending with tryptophan. Okay? That we said was a metabolic pathway. And the steps, you go from precursor to substance 1, 2, 3, 4 before you get to tryptophan. Those sort of intermediates were produced through the catalysis of an enzyme. And each one of those enzymes is a product of one of the genes within the TRP operon. Right? And we said that TRP operon is all those genes are similarly regulated so that when tryptophan's high, those genes are off. We don't need to synthesize more. When tryptophan's low, the operon is on because we need more tryptophan, or the cell needs more tryptophan. Okay? Normally on to synthesize tryptophan, but it is repressible. Okay? And that's where we are today. We're going to go back over the TRP operon in a minute. It's a repressible operon. All right? A repressible operon. Meaning that it is normally on, but it can be shut off. Okay? The inducible operon is opposite. It's normally on, or normally off, excuse me. The inducible operon is normally off, but can be turned on in certain cases. All right, we're going to talk about examples of. All right, the lac operon is an inducible operon. It contains genes that code for the metabolism of lactose. All right. So normally E. coli cells, and these are mainly in bacteria, and we use E. coli as an example because it's the most well characterized. All right. Normally in E. coli cells, they use glucose as a energy source. All right. And we went over this on Friday. All right. Use E. coli as an energy source. That's the most efficient source of energy. We get the most amount of ATP from glucose. Okay, fine. Sometimes use an E. coli cell will use up all the glucose that's available in solution, and then what does it do? Then what does it do? All right, it's going to find something else. Okay? It's going to find something else to produce energy. Right? And one of those possible sources is lactose. Okay? In order to do that, it has to turn on the genes that are important in breaking down lactose, in metabolizing lactose. Normally they're off because glucose is there. And so why produce the genes that break down lactose when we don't need to? Right? So normally it's off, but when glucose is low or absent, the cell will utilize lactose as an energy source. All right? So, what is a repressor protein? And how is the repressor important in the regulation of these operons? You remember from Friday.
Turns it off or something binds to it and turns it on.
All right, so a repressor protein is sort of the regulator in turning on or turning these group of genes on or off. Okay? In some cases, we have to activate the repressor to stop the gene, or stop the synthesis of these genes. In other cases, we have to turn off the repressor to allow these genes to be turned on. So it really is, the repressor protein in a way controls the on-off switch. Okay? In the case of the lac operon, it's normally off. So where would you find the repressor protein? In what state would you find the repressor protein? It'd be active and it would be bound to the operator. All right, now it's coming back to you. Okay, or else you're at least waking up. Okay? So if in the case of the lac operon that's normally off in normal growing conditions, the repressor is bound. Okay? So in order to turn those genes on, we have to do something. We have to inactivate the repressor in some way. Okay? When glucose is gone, lactose is present. All right? So in this case, the available energy source, lactose, right, is the actual inducer. All right? An inducer is a molecule that will inactivate the repressor. Okay? So, bacteria have evolved so that, they use those products or substances that are readily available to them as inducers or co-repressors, etc. In this case, lactose is the inducer, and it inactivates the repressor to turn the lac operon on. Right? Remember that, because that's important in a minute. All right? So for lac operon, the inducer is allolactose, or basically it's a form of lactose that enters the cell, right, as it gets sort of changed in shape. All right? And when that happens, the inducer binds to the repressor protein, inactivates it, and now those genes are turned on. Right? And so we call those genes that are inducible genes or inducible enzymes, those the enzymes that are coded by those genes are inducible. They can be turned on in the presence of lactose, right? And then, and analogously, the enzymes of the trip operon are repressible enzymes because they can be turned off, right? So we have a repressible operon, which is normally on but can be turned off, is the trip operon. And the lac operon is an inducible operon. It is normally off but can be turned on. Okay? Note, you'd be able to compare the two of those. Well, I should say contrast the two of those because they're quite different. Compare and contrast. Right, Ben? So, here's the lac operon. Okay? And then up top. And it's a little skewed because of the picture. But normally it is off. Okay? Because it's inducible, it's normally off and we can turn it on. Where is the repressor protein? Attached to the operator. And so when the repressor protein is attached to the operator, RNA polymerase cannot transcribe the genes. Okay? Lactose is absent, repressor is active, operon is off. Now, allolactose enters the cell, that's the inducer, and down here in green. Okay? It binds to the repressor protein, inactivates it. The repressor protein is no longer bound to the operator. Allows RNA polymerase to bind the operator and begin synthesis of the LAC-Y or LAC-Z, Y, and A genes. Right? Those genes that are important in the synthesis or metabolism of lactose, galactosidase, permease, and a transacetylase. All right, so normally this is turned off, but can be turned on in certain circumstances. It is inducible. Okay, questions on the lac operon? Sarah.
Earlier you were saying that the enzymes, they make something. Was that amino acids or?
That's for the trip operon.
Okay.
Okay, so that's involved in the synthesis of something. We're going to synthesize, from a precursor molecule, we're going to synthesize tryptophan.
Okay.
Right, and yes, that is amino acid. In this case, where they make enzymes that result in the amino acid tryptophan. In this case, for the lac operon, we make enzymes that are going to metabolize lactose eventually into energy. We're going to turn lactose into a glucose molecule. All right, so here it is again, sort of the inactive form and then the active forms. Okay? So the first statement up here that inducible enzymes usually function in catabolic pathways. It's pretty intuitive, right? Because what's happening? If an enzyme is going to sort of regulate the reaction of breaking something down, right? We're going to start with product A and we're going to break it into its smaller parts. Okay? If product A is not present, what's the point of producing the enzymes that do that process? There is none. It's an inefficient process, right? Why waste the time and energy to produce and transcribe genes that are not going to be used? Okay? But when something is present, such as lactose, and needs to be broken down, that's a way of inducing enzymes and making the process more efficient, only producing those enzymes, transcribing those genes when they are necessary. All right? So for the most part, inducible enzymes usually function in catabolic pathways, right? And their synthesis is induced by a chemical signal, an inducer. All right? On the opposite end, repressible enzymes or repressible operons. They're usually turned on and involved with anabolic pathways. Right? They're usually on and building something up until the point where you have enough of it. Once you have enough, there's no point, it's inefficient to continue to make more when you have enough of it, right? Or too much. All right, so they are usually repressed by high levels of the end product, and that's your feedback inhibition. All right, feedback, you inhibit the enzymes within the process, within the synthesis pathway, right? Because you have enough of this. Okay? Regulation of TRP and LAC operons involves negative control of genes because operons are switched off by the active form of the repressor. All right? So for the most part, in bacteria, we talk about two types of regulation. All right? Negative gene regulation, where things are turned off, or positive gene regulation, where things are turned on. Okay? Now, although the LAC and TRP operons represent different types of regulation in terms of they're either repressed or induced, they are both still in the category of negative gene regulation because the repressor works to shut the genes off. Okay? And it really is the difference between the two is what form the repressor is in at the beginning. Okay? Either it's active or it's inactive. And then you go from there. All right? Questions on the TRP and LAC operons. All right? We're going to get into another aspect of the LAC operon in a second. Okay? Clear as mud? Okay. So we've talked about negative gene regulation, turning things off. All right, well, how do we turn things on? Okay, so we've said that E. coli will preferentially use glucose when it's present. However, when glucose is in low amounts, there's an activator, right, which says a catabolite activator protein, CAP, is present, and it acts as an activator of transcription. So this is different from a repressor protein. It's not going to stop transcription. This is going to help enhance transcription to occur. Okay? CAP is activated by binding a molecule called cyclic AMP. All right? We'll see what cyclic AMP is in a minute. It's high when glucose is low. Okay? So CAP's activated, glucose is low. All right. Now, what operon you think is going to be involved here? Glucose low, probably the LAC operon. Okay. So, activated CAP, right, attaches to the promoter of the LAC operon and increases the affinity of RNA polymerase, thus accelerating transcription. Okay. So, in terms of the LAC operon, you actually have two things going on. You have a negative gene regulation where the repressor protein has to be inactivated, okay? And you have a positive gene regulation on the other side where there's actually an enhancement of activation of the CAP protein and enhancement of RNA polymerase binding to DNA. Right? So both of those things are ongoing. It's not just one or the other. Okay? So when glucose levels increase, what's going to happen? Is CAP going to be activated? No, because cyclic AMP goes down, CAP detaches from the LAC operon, right? Transcription proceeds at a very low rate. Okay? All right? Even if lactose is present. All right, so in the case, if I gave you an example of you're in the lab and you're going to figure out here's a new related E. coli bacteria, and you're going to see that it has the LAC operon present, and I give you a media that has both glucose and lactose at high levels. Okay? What is the transcriptional state going to be for the lac operon? Is the repressor going to be bound? Sarah says no. Why?
No.
Is the so what's going on today? Okay, someone stole my Apple TV. All right, so is glucose going to be bound? Yes or no? I mean, is the operator going to bind lactose or being bound by the repressor? Yes or no? Let's rewind here. So if I gave you this, let's minimize this. So I gave you, here's a flask, right, filled with E. coli, and in here it has high glucose and high lactose. And I asked you, what's the transcriptional state of the lac operon going to be? Okay? What questions do you need to ask yourself? So let's see. Lac operon, question mark. What questions you need to ask yourself? So there's plenty of glucose there. So in order to understand the transcriptional level of the lac operon, what do you have to know? You have to know the presence of glucose and lactose. I'm giving you they're both at high levels. So what do you expect is going to happen to the lac operon? It's going to be inactive or active? Okay, it's going to be inactive. You say transcription rates would be low or high? Low. Why would they be low? Because it prefers glucose. Okay. All right, so let's get to the reasoning why it's going to prefer glucose over the other. Is the lac repressor going to be bound to the operator? Why, yes. But lactose is high. So if lactose is high, lactose gets into the cell, lactose acts as what to the lac repressor? An inducer. So is that going to inactivate the repressor? Yes. All right, so remember we have two things going on in the lac operon. We have a negative gene regulation where it basically involves the action of the repressor. Okay? So if lactose is present, okay, that's going to stop the repressor protein. All right, that's going to inhibit the repressor. Inactivate it. Okay? But that by itself does not mean that the lac operon is turned on. Okay? Because we also have to have the other side of the coin. All right? We have to have the positive gene regulation that is ongoing. Because if glucose is high, what does that mean for the positive regulation side? Is CAP active? No. Cyclic AMP is low, right? So CAP is not active, so RNA polymerase doesn't bind to the promoter very well. Okay? So in this case, with high glucose, high lactose, transcription rates of the lac operon will be low. They won't be zero, they'll be low. Okay? What would happen if I said, let's get rid of this. Okay? What would happen if I said we had high glucose and no lactose? What's the state of the lac repressor going to be? Abby. All right, so the lac repressor will be bound to the operator, and the lac operon will be off. Why? Because there's no lactose, there's no inducer, so the repressor is active. So what's the transcription rate of the lac operon going to be in this case? 0. Okay, you see a difference there? All right. All right, so now the third possibility is, let's have low glucose, high lactose. What's going to happen to the repressor? The repressor protein is going to be inactivated. Why will it be inactivated? Because lactose acts as the inducer. Okay. What about CAP and cyclic AMP? All right, it says it's on or it's active because glucose levels are low. Glucose goes down, cyclic AMP goes up, activates CAP. So now what's the transcription rate of the lac operon going to be? High. Okay? So there's a couple, you have to know sort of the negative regulation, the state of the repressor protein, and that's in response to the presence of lactose. And you also have to know the positive end, right? And that's really the amount of glucose that's present. Okay? What would happen if we had low glucose and low lactose? Now what happens? So the inducer's still there. All right, you guys are thinking, this is great. The inducer's still there. Good. Okay? Cap is on. But what's the transcription rate going to be? Still low. Why? Because the amount of lactose. Right? Remember that it's a dynamic thing. You have to have so much lactose to bind all the repressor proteins that are present. Okay? So if lactose is low, transcription rate will still be low. Okay? Even though CAP is there, positive regulation's there, so there's a definite relationship between both sides of the coin here. Questions on positive versus negative gene regulation. All right? So CAP also helps to regulate other operons that are involved in catabolic pathways, so that when glucose is low, it's like an all-out blitz. Okay? We have to do something to make up for the energy deficit. Right? Whether it be, we have to find the cells looking for anything to break down and produce energy. Whether it be lactose, galactose, any other sugar, protein, whatever. So when glucose is low, all those other operons get turned on. Okay? So here's the graphic showing the two of this. All right? Promoter region, your CAP binding site, and your lac operon. All right? When it's cyclic AMP. All right? So lactose present, glucose scarce. All right? Cyclic AMP goes up. All right? You end up associating. CAP is activated, binds to the promoter, and stabilizes the RNA polymerase association, and mRNA is synthesized at a high level. Okay? The other end, lactose is present, glucose is present. Okay? Both of them are there, but only a little bit of lactose mRNA is synthesized. All right? Because that positive gene regulation portion isn't present. Okay? Questions on positive and negative gene regulation and the lac operon. All right, two more. All right, so that basically covers prokaryotic gene regulation. We talk about positive and negative gene regulation, the fact that the genes are grouped together in operons and under similar regulatory pathways, okay? And mainly, This all happens at the level of transcription. Okay, at the transcriptional level. In prokaryotes, most gene regulation occurs at the transcriptional level. You regulate the production of the message. All right? Once the protein's there or produced, then it is the way it is. Okay? That is a stark difference from prokaryotes to eukaryotic gene expression regulation. Okay? There are several levels, and I've given you the 4 different levels that you will encounter in eukaryotes. All right? So, in turn, what is this important for? Differential gene regulation. What makes a skin cell different than a neuron? Structure. What makes the structure of a neuron different from a skin cell? What makes a muscle cell different from a kidney cell or a liver cell? Why are they different? Let's go back and say, do they have the same set of code of instructions? If you picked out the cells from your skin and a hair, DNA from your hair, would the instructions still be the same? Dean says yes. Okay. So they still have this great big book of all the proteins that could be made. Awesome. So why is it that your skin looks, the cells of your skin look, act, behave differently than a neuron? Certain structures that were activated. And those structures are formed by what? Genes. So there's a different set of genes that are turned on or off that determine that you have skin cells versus neurons. And I specifically picked skin cells and neurons because they actually originate from the same area of the first cell that came about. So you start out as one cell, right? Yes? And you grow into 2, 4, 8, 16, 32, memorial stage. And then at that point in time, you get to be this big ball of cells. Other stuff starts happening. Cells start to move. Cells move, and they start talking to other cells, and then things start happening, and they start becoming different things. Okay? When cells start to move, you have all these cells on the outside of the embryo that are called the epidermis, right? Epidermal cells ectodermal cells on the outside. At some point in time, some part of those cells on the outside go back into the middle of the ball of cells and end up becoming your nervous system. Your brain, spinal cord, all the nerves of your body. Okay? But there are some cells that are left on the outside, still ectodermal, related to those cells that went in, that become your skin. The difference being is where they went, and then what genes were turned on and off in response to that. Okay? That's the importance of differential gene regulation. Why does a skin cell, because it stays on the outside of the embryo the whole time, have a different set of genes that are turned on than those cells, those related cells to it, the nervous cells that went into the embryo and started turning on different sets of genes? Why is that the way it is? Is because certain genes are turned on, certain genes are turned off. This is essential for what we call cell differentiation or cell specialization. Why a kidney cell is different than a muscle cell, and so on. All right? And we call that it's called differential gene regulation or differential gene expression. All right? And the differences between cell types. Right? The expression of different genes by cells with the same genome. All the cells of your body have the same genome, the same set of DNA instructions, right, that code for all the proteins that could be made in your cells. All right? Some genes are what we call muscle specific. They're only present in muscle. All right? Some are cartilage specific, right? Some are only found in certain tissues, right? And so this controls your development and how genes are different from how cells are different from one another, right? And this regulation or expression is regulated at several different stages: transcriptional, post-transcriptional, at the translational level, and post-translation. And on Wednesday, we'll go through several of those. Okay? Questions on that introduction? All right, and so you see here, there's an idea. Transcriptional level is everything up to and including the production of a mRNA. Right? Post-transcriptional, after the production of an mRNA, so that regulating export and RNA processing. Translational, okay, up to and including the formation of the ribosome, the formation of a polypeptide, and then afterwards, the action or lifespan of that polypeptide.